So far, we've been talking about neurons releasing chemicals and receiving the corresponding signals, and we've been purposely a bit vague about the details. So let's spend a bit of time going over the detailed machinery of the synapse. From this point forward, we'll simplify the terminology and say synapse when we mean chemical synapse. Since information transmission in a synapse is directional, we need to keep an eye on which neuron is presynaptic and which neuron is postsynaptic. The presynaptic neuron is where the action potential enters from, and the postsynaptic neuron is the one that receives the signal. The space between them is called the synaptic cleft, and it's usually only 20 to 40 nanometers in distance. This is a tiny distance, about 1,000 times thinner than a human hair. It might be tempting to think of this gap as a free extracellular space between the two neurons, but in reality it's a tightly regulated space whose geometry is governed by a complex infrastructure of adhesion molecules. Likewise, its chemical content is aggressively regulated by a phalanx of molecular machinery. You'll notice that as the axon of the presynaptic neuron gets closer to the synapse, at a certain point, it balloons out slightly. This area is called the presynaptic terminal, and it contains much of the specialized molecular machinery needed for the synapse to work. Here are the steps of synaptic transmission in broad strokes. First, an action potential travels down the axon to the synapse. When depolarized, the signal arriving at the presynaptic terminal introduces a release of pre-made neurotransmitters, or neuromodulators, into the synaptic cleft. These molecules are packaged into structures called vesicles, which you can think of as small hollow spheres that are made out of the same stuff as the cell membrane. They are somewhat like incredibly tiny little mini-cells, whose job is to hold a small quantity of signaling molecules at the ready. The arriving action potential causes these vesicles to fuse to the membrane and become part of it, releasing their payload into the synaptic cleft in the process. These molecules then diffuse across the cleft and bind to specific molecules called receptors on the other side of the cleft, on the postsynaptic terminal. A receptor is a molecular machine whose job is to bind to a very specific molecule, or class of molecules, and as a result affect some change in the postsynaptic neuron when they bind to the signaling molecule of interest. They're kind of like a little machine that can be turned on by a particular key. There are many different kinds of receptors, which can exert different kinds of influences on the downstream cell. Some receptors are themselves ion channels. When they bind their neurotransmitter of interest, the channel opens. These receptors are called ionotropic receptors. Ionotropic receptor channels can be selected for particular ions or groups of ions. As a result, it's not hard to imagine how one might depolarize a cell in response to a synaptic release. For instance, a channel that lets cations into the cell can depolarize the cell. Not all receptors are ion channels, though. Some receptors set off a complex chain of actions, molecular cascades within the cell, in some cases altering gene expression within the cell. These receptors are called metabotropic receptors, and they play an enormously important role in the function of the nervous system. We'll cover both ionotropic and metabotropic signaling in more detail in future lessons.